Hello everyone, welcome to our Ant Biology webinar series. I'm Shohali Vishwas, today's moderator. We are very much delightful to have Dr. Girdhari Lal as our today's speaker. To our audiences, you can post out your questions in the comment sections below and we will moderate them at the end of the talk. Continue to listen till the end to know more about the work the speaker is going to say. Hope you will enjoy the talk and get enriched with the talk. Our mentor and convener, Professor N.R.I. Banerjee, has conveyed her good wishes to our speaker. And now, without further delay, I would like to introduce today's speaker, Dr. Girdhari Lal, to our audiences. Dr. Girdhari Lal is a senior scientist working at the National Center for Cell Sciences, Pune. Dr. Lal obtained his PhD degree from the Indian Institute of Science, Bangalore in the year 2005. He was an assistant professor at Mount Sinai School of Medicine, New York City, USA before returning to India in the year 2010. Dr. Lal's laboratory is interested in understanding the cellular and molecular mechanisms of immune tolerance. His laboratory is investigating the chemokine receptor signaling and neuroimmune communication in the inflammation and autoimmunity. He has published more than 55 peer reviewed high impact factor journals such as Nature Immunology, Immunity, Journal of Clinical Investigation, Blood Journal of Autoimmunity, Journal of Immunotherapy of Cancer, Theranostics, and Journal of Immunology. He presented his work in more than 150 national and international meetings. Dr. Lal is also the CEO of the Ramalingua Fellowship and Innovative Young Biotechnologist Award from the Department of Biotechnology and the Swarna Jayanti Fellowship from the Department of Science and Technology, Ministry of Science and Technology, Government of India. He is a founder of Executing Council member of the Society of Inflammation Research and also Immune Oncology Society of India. Currently, Dr. Lal is also Vice President West Zone of the Indian Immunology Society. This is the introduction of our today's speaker. Now the platform is all yours, sir. You can start your talk.
Thank you. Thank you very much for very kind introduction, and also it's a pleasure to be part of your Sunday evening this iron biology lecture series. And thank you for inviting me. And it's honor to be participating and interacting with all of you virtually, although. What we decided actually to discuss today neuroimmune communication in health and disease, the gut feeling. So the topic what we are going to discuss about the what is known in biology is neurology and also now immunology. So in general, what is discussed actually independently, neurologists, neuroscience people working independently, and immunologists are working together, working independently and trying to understand what goes on these two regulatory mechanism in the body. Now the boundary is very narrow. Now we know that there are crosstalk between these system and how these neuroimmune communication happen and how they control disease. So I'll give you some flavor on this and gut feeling. What is special about neuroimmune communication in the gut? How that participate in the mounting immune response in the gut. So whenever we talk about immunology, we always discuss that the brain or central nervous system is immunocompromised site, that immune cell do not have easy access to it, and they don't go there. They don't have easy access, so that's how the immune response is not mounted against the neuronal system. But there are diseases, and in those diseases, in central nervous system, immune cells do go in and they damage the central nervous system, and we know a lot of disease about the neuroinflammation and neuroautoimmune diseases related with autoimmune response generated by the immune system. So when we look at the immune system, and since there is a barrier which provides the protection from the excess of the every each and every immune cells to access the neuronal tissue. And if you look at our brain, it's like 640-kilometer network of these blood vessels in our brain, and this much length of the blood vessels and blood is always flowing, and still we say that the immune cell do not cross these barriers and they actually bound to the territory. And when we talk about this, brain represent only 2% of the body mass but consume almost 20% of body glucose and oxygen. So now probably nature has created this way that you have to have this much blood vessels in a small volume, and the requirement is like that. And if we talk about capillaries, which account for almost like 85% of cerebral vessel and are the major site, these minor capillaries, that's the major site where the blood brain barrier actually work. And if you try this, open up this space and try, let's say, make a floor out of it, put all the cells in a plane surface, you will end up something like 12 meter square meter endothelial surface. So you have 12 square meter surface area, and then your immune cells is like 10 micron to 10 to 15 micron. So how many cells can actually access this area and try to evade? I think you can imagine now, with the length and the width of the excess barrier the body has to make it. And we also know that, and also not only that, the distance between one capillary vessels is almost not less than 40 micrometer. So intercapillary distance. So all these things nature has arranged in such a way and still maintain the boundary and integrity. But there are situations where the sometimes these barriers are damaged, and sometimes this damage is created by our own immune system. They produce molecules like cytokines or some other factor which is responsible for this barrier. So now imagine that when you have a fever, you also end up with headache and other response associated with that. So when in body responds to infection in the peripheral system, your brain also responds to it. And if you, that's one way of looking at infectious agent and how they are communicating. And now let's imagine that you have some kind of stress, some kind of something which is stressed or some other factors which is damage disturbing you. Your body also respond? Does your immune system also responds the communicate to each other? I think that's very interesting area to learn. And more importantly, this barrier what you have, how this barrier maintain integrity, how immune system talk to each other. If you look at this barrier, so you have endothelial cells lining, so one cell with other cells, they are connected with very tight junction molecules, and the immune cells are flowing all the time, and all kinds of immune cells and also the molecules secreted by them. Now, question is, if this barrier is damaged and if it's participating in immune reaction, which one and what they do and how they do? That's very, very interesting area, because if you look at cytokines, you have n number of cytokine. Is all of them are responsible for it? Is, if that is the case, do we have damage in all type of diseases? No. If answer is no, then who and what and how they damage it? That's interesting area, and we worked out that there are specific cytokines which are able to do this, but there are not all other cytokine which can damage this barrier, and those are the one which actually create trouble when we have serious infection. One of them is interferon gamma. So I'm not going in those all molecular detail. What I wanted to point out here is that you have molecules produced by immune cells which actually talk to these barriers and try to damage it. And if it damages, then the immune cells which do not have access under homeostatic condition, they also go in, and when they go in, they actually participate in immune reaction. All right, that's the talking about brain. Now, when we talk about gut also has a problem, and also the nature also build up the scenario that how the immune cells actually go there in the gut and how they stay and how they exit. So if you look at the gut also, you have epithelial lining, and the immune system actually always line up there. And the way they go, the way they decide from your secondary lymphoid organ, because they express a lot of chemokine receptors, and that's what these cells, because of their ligands are expressed by different cell in the gut, and the cells express these chemokines like chemokine receptor 5 or chemokine receptor 6, cytokine which is CCL20. They try to attract here because of this chemokine receptor interaction. So you have variety of cells coming in. So there is the movement of cells in the gut. There is a trafficking of cells when there is a blood-brain barrier is damaged. Now I will discuss only one of these to begin with. There are a lot of interesting things to learn, that how these cells decide and how they contribute to it. So if you look at some of these chemokine receptor, they actually bind to several ligands. That means chemokines, and they are redundant in function. And nature has created for a purpose. I think we can discuss that part later sometime. But what I wanted to say that let's discuss one thing, which is like here chemokine receptor 6 and CCL20. Now you have these two molecules, and when these molecules are interacting, how they interact, what they modify in the gut, what they perturb, that's the area, interesting area. So what is CCR6? Is a chemokine receptor which is expressed by a lot of immune cells, dendritic cells, various CD4 and CD8 T cells, CD1 D restricted NKT cells, and majority of the B cell in the gut. And the ligand for this is CCL20. And importantly, if you just one chemokine receptor, if you look at variety of diseases, including from autoimmunity to infection, HIV, or even graft versus host disease or cancer, if you have this chemokine receptor deficiency, knockdown, or the chemokine, the blocking of this chemokine, you actually control these all of these diseases. So what is suggesting to you that a chemokine which drives the cells migration to the to these organs, actually if you block them, they can actually control it. But that is true, is that the only limiting factor, that is only chemokine participating in the migration of the cell? No, beyond there might be beyond that function, because chemokine receptor are G protein coupled receptor, and GPCR signaling, when they bind to ligand, there are a lot of downstream molecules also get triggered. So we trying to understand that how these molecules participate, and to do that we use the animal model, and the animal model of gut inflammation or colitis. So the colitis can be induced in mouse a variety of ways. Two of them is mentioned here, like acute colitis, where you just take the mice, give 2% extra sodium sulfate in drinking water, and when mice drink this bleach, the epithelial barrier get damaged, and when they are damaged, the mice develop colitis within 7 to 10 days. And you can see that the mice actually have started losing the weight very abruptly here in this case. The other model, which is chronic colitis model, where you take immunodeficient mice, like one knockout mice, we do not have his own T cell and B cell, and inject fresh naive cells from another mice. Now these cells, once go in, they start damaging, localize into the colon, and then they actually start damaging the induced inflammation in the colon. But in same time, if you just inject Treg, this function of the cell which you have transferred is blocked, and mice do not develop colitis. So that one way you can test the function of variety of regulatory T cell in this model. But however, because these cells has to damage the barrier and the inflammation, it takes longer. That's why it's a chronic model, whereas this damage much fast, and then you have this colitis model. Now, what we showed that in human and mouse, that because it's signaling molecule, it stimulates a lot of this signaling cascade, and then it actually inhibit the function of regulatory T cells, and also promote inflammatory cell, which is marked by ROR gamma, which is called as TS17 cells or regulatory T cells. So essentially, just by modulating the chemokine receptor signaling, both in human and mice, it just alter the whole pathophysiology in the microenvironment where the epithelial cells are producing CCL20. All right, so let's move on to understand that now how neuroimmune communication contribute to it. So whenever we talk about neurons, we always talk about depression, suppression, or there is always a study with neurological disorders. So this is one of the study where it came in news, where people have what is published is that 43% of Indians are suffering from depression, and variety of depression mechanism, and 26% of them, and this is done with the in 10,000 Indian were surveys, and 26% with mild depression, 11% with moderately depressed, and 6% severe symptom of depression. So now you have neurological disturbance in the body, and now how this case, how the immune system will be responding, how the people's immune system will be responding under the face of various kind of stress or depression. That's another very interesting area. Last year it was published, funded by Bill Melinda Gates Foundation and Indian Council of ICMR and Ministry of Health and Family Welfare. And what you see here, actually, that's really alarming. The alarming is that the depressive disorder or major depressive disorder accounts for much more percentage in male and female, both gender or male and female. And if you look at depressive disorder, the state which are mostly called as little rich, they have more depressive disorder than the state which is supposed to be not economically well, but they are doing much better. So more you have funding or resources in terms of financial resources, looks like there is some link to it. Anyway, that's not part of discussion today, but what I wanted to point out that you have depression in different states with the variety of the region. And also when you have this type of depression, then the conduct disorder also changes. So now you see green change to blue and blue change to red, depending on how things change. All right. So, and also, if you look at this disorder behavior, then you have the gender biasness, male and female, and in the male and female, you see females are much more prone to it, either anxiety or others, whereas there is a difference between one or other, or they may not alter. All right. Now coming back to what is his reaction in terms of immune response. So if you look at in terms of immune response and diseases, what you'll find that there are a lot of psychological stress which is associated with autoimmune diseases. So you know that IBD, inflammatory bowel disease, irritable bowel syndrome, or psoriasis, rheumatoid arthritis, multiple sclerosis, diabetes, chronic pulmonary sarcoidosis. There are n number of diseases which are also known to be associated with some kind of chronic stress. So now you have this immune reaction, which are mostly autoimmune reaction, how the neurological events, what is happening in the body, how the immune cells are responding to it. That's very interesting area to learn. Now coming back to the stress and the chronic disorder, what I want to point out that you can read this in literature and the news that there is number of people who are suffering from this type of depression. And in India also 13.7% Indian. This is old data which I showed it 2020. That's 27-28%. That's the recent data. And now what actually I wanted to point out, depressed people have increased level of inflammatory cytokine, which is IL-6, TNF-alpha, interleukin-1 beta, C-reactive proteins. Now you have some kind of association that whenever you're the neurologically disturbed, you also have inflammatory cytokines, and if you have chronic inflammatory cytokine around, then you saw the disease what was happening. Now another example I'll mention here, that if you treat an individual with a drug that produce inflammatory effect, that lead to depression. So the people who was actually promoting their inflammatory reaction, they get depressed. Whereas when a person is treated with anti-inflammatory drugs, their depression is also improved, decreased. So there is a link between immune reaction and also neurological response to each other. And I can mention another example here, like in case of IBD. So in case of inflammatory bowel disease, which happen in the gut, we all know. But before it happens, your body also respond differently, either your skin, mouth, eyes, bones, and every other place, that before extra intestinal manifestation, before and after as well. So and most of these symptoms appear either early, some of them you see that they at later stage, But what is these connections? Why actually something happened in the gut, the your other body part are responding to it? What is association, and how and who is carrying this information from one location to other, and that's how you can have it either after or after these diseases. That's interesting area to learn. Now let's talk about more on immune system in the gut. So you know that immune system in our gastrointestinal tract, the first thing starting from the mouth, you have a tonsil, and tonsil has lot of immune cells and one of the secondary lymphoid organ actually. And now, if you look into the intestine, you see the small white patches all around the intestine. These are called Peyer's patch. And if you look at more detail about this patch, you see there is organized structure here. Very well organized structure, which is present in the lining of the gut lining. And if you look more details, you will find that there are T cells, there are B cells, a lot of cells actually, and they are arranged in a specific manner. So a lot of these cells are present at your barrier site, which are either producing cytokine, and always because we know that our gut are exposed to the external environment, which are pathogenic or even dangerous to us. So probably the nature has created this type of intestinal sentinel that you should have these pairs patches around and keep on sampling and responding to it. Perfect. Now let's talk about more in terms of gut. So if we talk about gut, what people call it a second brain. So what you have in the gut, how you respond to your gut, I mean it senses all part of your body. The reason for it, that if you look at just your gastrointestinal tract, you have almost like 5 meter length of your intestine, and almost in everybody, right? And if you look at, if you put all these 5 meter epithelial barrier what you have, which is monolayer, that's even more interesting, unlike what you have in skin with multi-layer. And if you put this intestinal barrier in the surface, epithelial surface, you will end up with something like 32 square meter surface area, which is much bigger size of your room. Now if you have this much barrier, again 10 to 20 micron cells, how many cells you require to guard, and the size of bacteria, you know very well, right? So probably that is why nature has created that about 70 to 80% of your body's immune systems lies at the gastrointestinal tract, and almost 100 million neurons actually sensing all the time what goes on around it, and around 100,000 intrinsic nerve ending, and 40 trillions microbes are there in our gut. So and variety of them actually, not one type. So all these things, they have to protect us from the invading microbes, and also responding to it, they require this type of assembly. So that's nature smartly has created the second brain, which is gut. Now what it happens, how it happens, that now to understand more details, the regulatory mechanism what operates is like immune system, you have nervous system, and then you have endocrine system. So all these systems has to work in a homeostatic manner, in a way that if you disturb any one of them, either nervous system, your immune system, and endocrine system is disturbed. If you disturb this, your other system is also going to disturb. So it has to be having synchronized, and they have to talk to each other in order to have synchronized function. If they don't talk, they're not going to operate each other very well. So now the question is how they communicate, how they talk to each other. That's interesting area to learn, and probably if to understand that, probably gives a new area to understand the biology of different diseases. Let me put to you one example where you have nerves. We know that neurons. If you look into nerves, they have a lot of these neurotransmitter signals, obviously, because that's their job. But if you look more closely, the nerve also has a lot of receptors which are immunologically important, like interleukin 1 beta, TNF alpha, IL-6, IL-17A. All these are immunologically important. So if anything happen in the microenvironment, the immune system will respond. They actually, these nerves sense it, and once they sense, they actually definitely transmit the signal from one area to other. So now you have another way that when the nerve fires, some kind of neurotransmitters. The immune system, if you look closely, which is not mentioned here in the slide, but I can tell you, they also have lot of receptor for these neurotransmitters, and how they sense, how the immune system responds. That's an interesting area, and that's where our lab is focusing on to understand the biology of this neuroimmune communication. Now I'll take you one example where you can just appreciate that how just neuronal signals talk to immune system. So the experiment is very simple. So you have sciatic nerve which is draining to the lymph node. What is done here, the adrenergic receptor which is going and sampling the lymph node, and if you just disconnect this surgically, disconnect the nerve which are sampling the lymph nodes, and then ask question what happened to the immune system in the lymph node which do not receive this communication. Now on the same scale you see that this is unconnected, undisrupted, and this is a disrupted lymph node. Now you see your lymph node is much different than what you see in the normal condition. So just by lymph node stop receiving the signal, you can see all other parameter of different cell types is actually, just by grossly looking, you can appreciate that just by not receiving a signal which is supposed to be neurologically important adrenergic signal. You know all about this adrenergic signaling and its response. If you don't have, your immune system is disturbed. So that's one example that how the neuronal signal controls the immune system. Now let's understand this in more detail and then try to understand how this works. So in biology, we know that the nerve fibers connected each other, and when stimulus comes from one side, pass on to the other side through these neuronal network, right? And depending on from where the stimulus is received, like skin, and who is the target organ to respond to it, like muscle. Likewise, the inflammatory reflex, you have neuronal connections where secondary neuron, the interneuron connection, vagus nerve, and these nerves are finally also connected to the secondary lymphoid organ like spleens. Now, when these nerves fires, they produce neurotransmitter. You have immune cells which are actually having the receptor for these neurotransmitter. One of them is acetylcholine. We know acetylcholine as a neurotransmitter. And when the nerve cells fire, the immune cells actually secrete, as well as nerve cells secrete this neurotransmitter. There's a response in terms of reducing the cytokines or altered cytokine response in the secondary lymphoid organ just by giving this inflammatory reflex. Now let's understand that how does it really work. So I'll give you one example here. And the experiment is done is here in a way that you take a mouse and you just perforate, just cut the intestine. So what will happen if you physically damage or perforate the intestine? All gut microflora will come out, and then this will have peritonitis or the sepsis kind of condition, and the mice will die in this way with time. Now, what you do here, you disconnect the nerve, vagus nerve, which is going to the spleen, and do the same experiment, perforate intestine, and then ask question, what happened to survival of mice? Only disconnection in the nerve which are going in the spleen. Now you see survival is drop, right? Now you can do the experiment differently. Instead of cutting this or disconnecting it, you can electrically stimulate the same nerve and then do the same experiment and ask question, what happened? What happened to the survival of mouse? Now you see when you stimulate it, mice is surviving better. You can do this by, instead of physical, you can do by chemical antagonist or agonist, esterase inhibitor or using agonist. And if you do that, survival is improved. What is telling you? Just by neuronal communication, altering the neuronal communication, the immune response to infection and inflammation is altered. And that's why probably that is now is used in arthritis, and there are other places people are trying to understand that how we can stimulate these nerve signal which also contribute in controlling various inflammation. So that's very interesting area to learn how these neuroimmune communication work in our body, how they talk to each other in order to protect us from various outside invader or even from autoimmunity. Another example I'll take here, which is acetylcholine. We know it's acetylcholine molecules. It's choline and then the serine molecule with different biochemical reaction. Choline acetyltransferase makes this acetylcholine, and then acetylcholine released, and this acetylcholine can bind to the various receptor, which is muscarinic acetylcholine receptor or nicotinic acetylcholine receptor. So when we look at these, and there are enzyme in the cell, on the surface of cell, which is like esterases, which degrade the acetylcholine, again put back into the acetic acid and choline. Choline goes back recycled. So this is kind of metabolism of the acetylcholine. But interesting thing, what I wanted to point out, that not only nerves, actually immune cells also produce acetylcholine, and they also have receptor for them, like muscarinic acetylcholine receptor or acetylcholine. The difference between the what happen in the immune cell versus neuronal cell, the neurons actually can store acetylcholine and when they can trigger when they need. Whereas in mouse, in the immune cells, the moment they are synthesized, they're thrown out. So they have to sense by someone in the vicinity. All right, just keep in mind. Now, in terms of receptor, when muscarinic receptor, which is GPCR, or ion channel like the nicotinic receptor, both of them are present on CD4 T cells. And when the ligand bind to the receptor, and when which happens that these are immune cells, because we know GPCR, and then they can actually fire different signaling. Which one works where, I think we don't know. We need to learn about it so that we can understand how these these neurotransmitters, when they act on immune cells, how they modulate. I'll take some example to prove that, to show you that how interesting it is. So you saw that acute colitis model where mice were fed with distant sodium sulfate, and if you feed them distant sodium sulfate, the epithelial lining is damaged, and the mice will not able to absorb food, and then you will have loss of weight. But now what you do here is that, if instead of DSS treatment, you can also, I think that is missing. If you don't do anything, that's control, mice will keep on gaining the weight with time. Now in this case, you put antagonist here, which is atropine, and there is another antagonist. And if you do that, the mice, instead of waiting, losing the gain, now they started regaining the weight. So just antagonizing the receptor, MSHR antagonizing its effect with atropine, now you see that the mice actually stopped losing the weight with time with DSS. And if you go back and see what happened in the mouse, inside the mouse, you'll see that the mice actually, when it treated with distant sodium sulfate, the regulatory network, which is marked here with FOXP3, they upregulate. And the inflammatory cell like the ROR gamma T, which marks for TS17, their frequency is reduced. So by altering this, by chemically altering, actually the inflammatory and regulatory immune cells disturbance get disturbed and probably provide protection. So that's another way to test that. You take the cells in petri dish, because when you're giving the antagonist in mouse, you don't know what is working with what. But you can do in petri dish, and when you can take purified cells and just throw this antagonist and ask them in the differentiating condition that let the T cell differentiate into Th1, Th2, or Th17, regulatory cell or inflammatory cells, and presence or absence of antagonist like atropine. And now you see that Tregs, when exposed to atropine or antagonist, they increase their frequency, whereas the frequency of Th17 cells drop down. So inflammation inducing cells are growing down, whereas regulatory cells going up just by antagonizing one of these receptors. Perfect. Now I'll just take another example, serotonin. Serotonin, we know, happy hormone, and I'm sure most of you know that the importance of this hormone, keeps you happy. And if you look at the biology, tryptophan, in terms of biochemistry, tryptophans, by using the tryptophan hydroxylase, get transformed, and then serotonin. And serotonin acts on various cell type. In fact, platelets are major reservoir. Now, and endochromaffin cells is the one which synthesized in the peripheral system. And also you should know that serotonin is also a neurotransmitter and also a hormone. But the serotonin, what is produced in the brain versus what produced in the peripheral system by endocrine cells, they might work differently. All right, and if you look at the more biochemistry of it, that you have different types of serotonin receptors, 5-hydroxytryptophan, serotonin, it binds to receptor, transmit various signal, and let's say the lot of immune cells actually express this receptor. So now you can ask question, what happens when serotonin is bind to the immune cells? Does immune cells actually respond to it? So in this case, B cell, we know that B cell produce maturation, NK cell degranulation happen, dendritic cells, antigen processing, presentation, macrophage differentiation. There are a lot of other pathways which the just signaling from these neurotransmitters within the immune cells can get altered. And not only that, there is recently reported, I mean, not recently, 2019, that uh, just gut microbiota composition is modulated, altered if you just have serotonin signaling, can influence and also the intestinal response and susceptibility to colitis just by altering this one of the neurotransmitter which is here. Okay, microbiota, and again, it communicate with the neuroinflammation and the different axes of neural signaling. And in this case, that's what I was trying to tell you, that when you have epithelial signaling, responding vagus nerve responding, then the short chain fatty acid produce, and then other kinds of immune response. And in all those cases, the 5-HT or serotonin gut bacteria can actually modulate the whole component, and then it can have effect what's going on in the gut, which is endochromaffin cells were producing this 5-HT. They can influence gut microflora, and if they can produce the various gut microflora is altered, then the metabolite produced by gut bacteria may be different now. And if that is different, they might activate different signaling pathway, and that's what we see. Altered gut microflora leads to various diseases. And now there are several probiotics, and there is a clinical evidence associated with that. If you have the gut microflora, the altered gut microflora, the specific gut microflora as a probiotic, they have different ability to act at different system, including from antidepressant and anxiety-like effect in adult, or cognitive processing, and a lot of other information is known now. They have behavioral outcome to physiological outcome just by altering the gut microflora. To summarize this, what I told you, that you have a brain, you have a different signaling mechanism both in the gut and gut associated lymphoid tissues, and then you have microbiota communicating to it, and all of these has to link each other, and if you just altered one of them, it has consequence on our body, and that may have a different effect the way our body and immune system responds to it. So let me just start thanking then here the lot of students here. Those some of them has finished PhD and gone, and some of them are still continuing to try to understand how these neuroimmune communication work in different inflammatory diseases, how they respond to it, and also the lab is supported from funding from different areas. And I welcome most of the students to join, participate in the ongoing research in NCCS. And when there is opening, I think there is a very interesting science going on in NCCS, and we look forward to all of you. Thank you.
Thank you, sir. It
Yep.
was very enriching talk. Uh, Sir, there is no such question, but sir, I wanted to ask you one thing,
Mhm.
that you just have presented the slides regarding nervous system or neurohormone stimulation on the immune system, gut immune system. But
Sure.
have you seen any effect of interference of intestinal epithelial cell? Because epithelial cell and immune cell of gut or in the of the intestine plays a important role, or they interact with each other. So have you
Mhm.
seen any effect through giving some neurohormone stimulation, and by that through the interaction of neurohormones, have there any effect of intestinal epithelial cells as well?
Right. So we have not directly looked at epithelial effect of these hormones on epithelium, but I think the reports are there in the literature that it alters the chemokine pattern. It alters also the various other metabolites. which actually participate or which attracts the immune cells into the gut. So there is known interaction, which one of the example which I showed to you, that serotonin, just by altering the epithelial integrity, it alters the gut microflora as well. So there are a report in literature which actually says that there is neurotransmitters which actually act on these epithelial cell and alter their physiology and function.
Okay. Thank you, sir. We have question from our audience. Uh, is there any role of neurotransmitter in depression?
So, Ritam, I will say that why did you get depressed? Is it because of your gut or your neuronal system response to neuronal system? So I think what is happen is that there is a somewhat the disturbance in the neurotransmitter, the communication between the neurons or some organs, that's what leads to that undergoing in depression condition. If you are happy, the correct neurotransmitter which is fired will keep you happy. Serotonin is one of the example when it is fired in the brain, but it's not always good when it is fired in the gut, because it has totally different effect in the gut versus what you see in the what happen in the brain. So yes, there is a very good relation between depression and neurotransmitter.
Okay, sir. We have the second question. Uh, Cytokine storm during COVID, can it give physiological symptoms considering the pathways you discussed?
Very good question. So Chaitali, I will say that when you have this cytokine storm, and like I showed you the example that the neurons are expressing different type of receptors, and these receptors do respond to it at different places at different capacity. Now I'll divide this to understand more better. I'll just divide in different sections. Let's say, for example, you have blood brain barrier, and the endothelial cells on the blood brain barrier, they also have receptor for these cytokines. And when you have too much cytokines, and it so happen one of the cytokines which participate in damaging the blood brain barrier. Now, if your blood-brain barrier is damaged in presence of this sudden shock or cytokine storm in the body, you know what are the consequences your neuronal system is going to face, right? It's going to have super pressure on the neuronal system, because now everything fluidics are damaged, and then you say that stroke, also one of the characteristic you see that sudden death in the patient. Other than that, in terms of talking to neuron, now going, let's say cytokine went in and then now neurons, and because neurons are responding to these cytokines, they will definitely have altered function. And then you know that neuronal signaling to control various organs, including the vital organs, may have effect. So cytokine storms, that's why have very prompt and very adverse effect than anything else.
Okay, sir. Uh, Now the next question is, what is the effect of serotonin in gut? Is it good or bad?
Okay, so very good. So, serotonin in the gut, that's a fantastic question. So, one of the example which Navinita I gave it, that if you have too much serotonin, it can alter your gut microflora, and the gut microflora can be a positive one. And in this case, it alters and alter in a way that the symptom what you see in terms of originating from gut is colitis, right? So if you have more firing of serotonin in the gut, you will have the colitis response altered. Now I'll tell you another example. That's a very interesting question. It just came recently in Journal of Clinical Investigation in December issue, the very recent. So serotonin, when actually produced in the gut, obviously it circulates in the blood, right? It goes in circulation. Now you see that the muscle express one of its receptor called 5 the STR2B, 5 STR2B. Now when these receptors are up, it actually make more insulin muscle, more insulin resistance. So when the serotonin in peripheral system is more firing in making it more the insulin resistance, what is going to happen? You're going to have like diabetes, because hyperglycemia will come up because insulin is not responding to it. So now it's a happy hormone in the brain, not in the gut. I'll take another example to tell you. Uh, if you look at the colon cancer patient, and if you take the colon cancer patient and divide into two groups, because now sequencing data is available, you can go ahead and look into it. And if you just have these two patient divide into two category, one, the one which is expressing one of these receptor, and the one which is not expressing this receptor, colon cancer patient. And then ask question, those who are expressing very high serotonin receptor, how they are surviving, because those data are available in the database. And those who has less expression, how is their survivability in terms of the cancer progression and survivability. What you find is that when they're expressing high serotonin receptor in the gut, in the colon cancer, the patient survivability drops fantastically, very very low. That means they don't survive much. And that's what we did actually recently experiment. One student did experiment to find out why this is so. And when you antagonize this receptor, actually you modulate the immune response, and in fact it works as way anti-cancer agent. So just by modulating the serotonin receptor, actually you alter the whole physiology of the colon cancer in the gut. So it does respond. And also keep in mind that serotonin has various receptor, and receptor on different cell types varies, and it's not necessary what it works in one tissue or one cell, it may have the similar effect on other cell, because it may have different. We need to learn more about this signaling. But in gut, you now know that what are the things it can do.
Okay, sir. Thank you, sir. Uh, these were the questions.
Perfect.
These were the questions from the audience, and thank you, sir, for giving such a good lecture to us. And okay, another question came up. Do other hormones like estrogen also affect this pathway?
Very good. So the hormone estrogen definitely affect the immune system. There are immune cells which actually express this receptor, including macrophages and regulatory T cells, and they do respond to it. I'll give you one example. I showed you that especially in certain hormonal disbalance, in especially in the girls, you will see that face will have some pimple or some kind of skin disturbance on the faces. It's just simple hormonal disbalance. And the inflammation is created not by the skin epithelial cell, it's the your immune system which is going and responding on the skin. So and that is because of some of these hormones. So the hormonal connection and the immune system, they are definitely talk to each other, and slight disturbance in them will have consequences, which be one example which I gave is the small pimples coming on the face just by altering the hormonal disturbance.
Yes, sir. It is quite true. Thank you, sir.
All right.
Okay, sir.
Perfect. So thank you very much. I hope you will follow what I discussed today, and
Yes, sir.
thank you very
Yes.
much. Yeah, good night.
Thank you, sir. Good night, sir. And now, to our audiences, continue to listen to our talks, which are archived in our YouTube channel, and stay tuned to listen to other lectures which will be coming up in our next webinar sessions. Thank you. Until then, stay safe and goodbye.